Hi guys, we're going to be talking about glycolysis in a little bit more detail. If you haven't seen my first video in this series on the overview of respiration, I really recommend that you do that. I think it's a bit more important that you appreciate the overview of the process, how the three parts, glycolysis, Krebs cycle, and then finally oxidative phosphorylation and the electron transport chain. It's really important to appreciate how they work together and how they how the products of one kind of feed into the other, and it really helps to appreciate if one stage could stop for any reason, how it might feed back on the other processes. So do watch the overview first. I'm really not going to be adding too much information beyond what you have in your textbooks, but as usual, I'll just be trying to explain maybe why some of these steps happen. Okay, so let's begin. As before, it's really all about starting the process off. We're starting with glucose in the cell. It's a respiratory substrate. That's an important phrase. Respiratory substrate refers to any molecule that can feed into the pathway of respiration at any point to ultimately yield ATP as an energy storage product. Okay, so glucose is just one example of a respiratory substrate. Other things can enter. If you're starting with hexose sugars, it's not just glucose that can feed into this pathway. Other hexoses can also feed into the pathway. Okay, so let's begin then. So we're starting with glucose, and the idea is that we want to break this down ultimately to break this more complex molecule with more bonds, more atoms, and we want to break it down step by step. And as we go from this kind of larger molecule to the simple molecules such as carbon dioxide, as we go from more complex to less complex, we're going to release energy along the way, and essentially respiration is just a process to kind of break this, possibly a kind of very quick process into a series of stages where small amounts of energy can be captured and used to make ATP. That's the whole point. Because if this was all to happen in one stage, the release of energy would be quite explosive, really. And the idea is that the processes in the cells, they need quite small amounts of energy. So you don't want to be causing an explosion every time you want to transport something against a concentration gradient. What you need is to break this process of glucose to carbon dioxide down into many stages, each of those stages releasing a little bit of energy each time, and we collect this little bit of energy in ATP, and when we need those small amounts of energy, we break ATP down to ADP, releasing that small amount of energy, driving those energy requiring processes. So this is the beginning of that, okay? And it really only takes us a third of the way through. We start with glucose, which is a 6-carbon molecule, and we end up with pyruvate, which is a 3-carbon molecule. So even in this stage, we do quite a lot of breaking down, and but essentially, the main product of this process is pyruvate. Okay. Yes, we do make a little bit of ATP. Yes, we do make a little bit of reduced NAD, but the main thing that we produce is pyruvate. So we're going to look at that journey. Remember, if you've seen the overview or you've come across this before, pyruvate is what then goes into the mitochondria to drive the Krebs cycle, and the Krebs cycle is what generates the large majority or the large proportion of reduced NAD, reduced FAD, which drives the electron transport chain, which drives oxidative phosphorylation. So all of that stuff can't really happen without the generation of pyruvate first. So let's look at that now. So we start off with this 6-carbon molecule in the cell, okay? And glucose can go in and out of cells quite easily through glucose transporters. You might have looked at glucose entry into liver cells, hepatocytes upon insulin signaling. Glucose can enter cells through the glucose transporters that are in the cell surface membrane of hepatocytes. But it can, if the concentration of glucose is higher in the cell than outside, it can easily go outside. So how do you retain glucose in the cell? Well, one of the processes is phosphorylation. If you add a phosphate group to glucose, it immediately then no longer can fit in that channel. So glucose that you have in the cell cannot diffuse out. So this is one of the key steps in glycolysis. It really commits glucose to this pathway, because once it's been phosphorylated, it cannot exit the cell. It's not complementary, it doesn't fit in the glucose transporter because it's got this huge, well, not massive, but it's got this very heavily charged polar group in the form of a phosphate, and it prevents glucose from then fitting into the channel that will allow its facilitated diffusion out. So this step is important. It commits. So this first phosphorylation, it commits glucose to the process of glycolysis. Okay, commits glucose to the process of glycolysis by adding a phosphate group. Okay, and remember, the overriding thing that you have to remember about all the processes that we're going to talk about is that we're really only looking at the substrates and the products. We're kind of ignoring that there's important enzymes that are carrying these processes out. Every single chemical change would not occur without an enzyme catalyzing it. So it's really important that we appreciate that these are enzyme-controlled reactions, and that we appreciate that this glucose is the substrate for the enzyme whose product is that. So that arrow is very significant because it means that there's an enzyme whose substrate is glucose and whose product is glucose 6-phosphate. And that counts for every single process here and in Krebs cycle. Okay? So it might be something you want to bear in mind for those odd questions that kind of ask slightly out of the box questions, such as how temperature might affect this process, how pH might affect this process. If those things can affect enzymes, it's going to affect this pathway, yeah? Because it's really that substrate coming in for that enzyme. That enzyme produces this product. That product happens to be the substrate of the next enzyme. Okay, and that enzyme produces this product, and that product happens to be the substrate of the enzyme that produces that. So all these chemical changes carried out by enzymes, we cannot forget that, even though we're going to not focus on the enzymes too much. I think it's more important to look at the modification, the metabolism of these molecules, and why that happens, and how that fits into the whole process that ultimately leads to ATP generation. Okay, so we've phosphorylated glucose, and we've said that it kind of commits glucose to this pathway. It gets phosphorylated again, and actually what I should mention, where do I leave my pens? Okay, I should mention that phosphate group actually comes from an ATP, okay? So there's an ATP that's just lost its phosphate group in this process, okay? So one ATP there, and glucose 6-phosphate undergoes further modification, but significantly another phosphate group is also added. So another ATP is used up at this stage, converted to ADP. Okay, so what we are looking at here is an addition of a phosphate group there, sorry, not there, here to generate glucose 6-phosphate. Further modifications happen, and in general we call this molecule hexose bisphosphate because it's got two phosphates. Interesting fact, just learned this today. Why isn't it called hexose diphosphate? Well, diphosphate implies that you've got one phosphate group and another phosphate group is attached to the first phosphate group. Triphosphate, the third phosphate group attached to that phosphate group. Bisphosphate implies that the phosphates are added but at different positions in the molecule. Never too old to learn. Anyway, so we've got hexose bisphosphate. Now this is kind of important because what these phosphate groups do is they destabilize the otherwise very stable glucose molecule. This glucose up here, very stable. And the problem with stable things is they don't react. Like, gold, silver, those kind of metals, the reason why they are popular for jewelry is because they don't react much. They have a very low tendency to react and therefore they keep their shininess. Same thing with glucose, almost, though completely different at the same time. With glucose is quite stable, so why would it bother to go through all this metabolic change? It's quite stable and happy as it is. It would take a lot of energy input to make it go through all this change. But the addition of these phosphate groups, first one there, second one there, to convert it into this, destabilizes it. It destabilizes the molecule because it's attached, these very kind of polar, negatively charged groups to that molecule, and it makes it more likely to undergo these very drastic chemical changes that are going to follow. So this is another important point about glycolysis, is that initially you'll see that we are using energy up. We are using ATP in a process where ultimately we want to produce ATP. So, and that's because glucose is quite stable. And if we want to destabilize it, we're going to need to put some energy in to make it undergo this change. All right? So that's kind of explains why you need to put energy in it in the first place. But once you've made this, that's essentially the kind of first stage over. And it's important to note that in the first stage of glycolysis, we have used up 2 ATP molecules. So -2 ATP. Okay, and that's important. But let's move on to the next stage now. Hexose 1,6-bisphosphate, i.e. hexose is phosphorylated on carbon number 1 and carbon number 6, gets broken down to 2 molecules of triose phosphate. Okay, so we should put that phosphate group there, and just keep in your mind that there are 2 molecules of this produced. 6 carbons, and so therefore we get 2 times 3 carbon molecules. Okay? Next stage. Triose phosphate is then converted into an intermediate. Now, I could go into details here, but really it's been simplified already. And for me, this could even be simplified further, but it's not. We need to know some of these things, so I'm going to stick to these details, right? But you will do better if you understand the generalizations, understand, summarize these processes, see what goes in, see what goes out. And once you are happy with those details, then delve in a bit further. Okay, so anyway, we've come to triose phosphate. Triose phosphate is then converted into an intermediate. And in this process, 2 important things happen. My arrows are a bit small to show these 2 very important things, but anyway, I'll do my best. The first thing is that we actually now get the production of an ATP from an ADP. Now that's significant because up till now we've only used ATP up. But this conversion here releases enough energy to be captured and formed, or to add, that energy is used to add a phosphate group onto ATP and store that energy for later use. So this is an ATP molecule that could be used somewhere to, I don't know, for active transport, or for the synthesis of a biological molecule somewhere. The second important thing that happens here is that we get the reduction of NAD to NADH. So the first thing is we get the synthesis of an ATP molecule, and at the same time, in the same process, we get NAD reduced to NADH. Now these are important. What happens to ATP here is called a substrate level phosphorylation. It's a formation of an ATP molecule that doesn't rely on oxidative phosphorylation. That's going to happen later. It's a phosphorylation event, it's an ATP synthesis, ATP formation event that doesn't rely on ATP synthase and the proton gradient and all of that. So this is called a substrate level phosphorylation, and you should be very aware of its kind of difference from the ATP that's formed at oxidative phosphorylation stage. Okay, and the other thing that happens is that we've removed hydrogen from this molecule and transferred it onto the coenzyme NAD. All right, so what we're doing now is we're not just looking at this as something that we need to remember. We're kind of looking at the explanation of what's actually happening. And I appreciate that it's difficult because you don't need to know the structures of all these molecules, and that makes it difficult to see the hydrogen moving from there to over here. But believe me, it's better this way, okay? By all means, if you do want to go into the details and find out about these things, do so. If you have any questions about that not making sense, you can ask me, no problem. So, hydrogen is transferred from the triose phosphate onto the coenzyme NAD, and therefore we get NAD reduced to NADH. Whenever something's reduced, something else must be oxidized, okay? So that's how it works. So because NAD has been reduced, because it's got hydrogen now, the triose phosphate has been oxidized, right? So triose phosphate is oxidized into the intermediate, but remember, this is happening twice because of our two triose phosphate molecules, all right? So at this stage, we have got plus 2 NADH and plus 2 ATP, just at this stage right here. Okay? Right. Now, the intermediate then gets converted into pyruvate, and again this happens twice for each glucose that goes into the glycolytic pathway, and here again we can get the generation of another ATP molecule from ADP. Okay, so ADP again, phosphate group is attached to that, or this conversion from intermediate to pyruvate has enough energy released during that reaction that we can join a phosphate group onto ADP and capture that energy to be used somewhere else. Okay, and again this happens twice. So put these arrows there. And this happens twice, so here again, so we should be asking ourselves, well, what is this? What do we call this event? We call this a substrate level phosphorylation. Okay, here again, so we have plus 2 ATP at this stage. So overall, what's happened during glycolysis is we have 2 pyruvate molecules produced from the single glucose that went in. We have 2 ATP used up in the first part of the glycolysis process. Remember, we had to give glucose a little bit of a nudge so that it was ready for all these further modifications. Otherwise it was too stable, so we had to put some energy in, and that came from 2 ATP molecules used up. But subsequently we produced 4 ATP molecules, 2 there, 2 at this stage. So 4 minus the 2 that we used, a net production of 2 ATP molecules, and we produced one reduced NADH here, 1 reduced NADH there. So we produced 2 reduced NAD coenzymes. Okay, so what's going to happen here? Well, this ATP just contributes to the kind of overall net production of ATP. The pyruvates are going to go to Krebs cycle, and the NADs will be transported into the mitochondria, where they will then pass on their electrons into the electron transport chain for oxidative phosphorylation. Okay, so I hope that's kind of explained a few things, and I've probably explained a few things, but also raised many questions, and maybe that's how it should be. But unfortunately, sometimes we come to processes which we could go into more detail, but it's really saving you a lot of kind of headache summarizing it like this. Remember, this is one of those processes where it's really beneficial to know the general processes rather than the detail. I know the detail will help. You might get a 1-mark question over there or a 1-mark question over there about state the enzyme, or describe this process or whatever. But the real explanation, the suggest questions will require you to have a broad understanding of these processes. This is one part of the bigger process. Only go into the details when it helps you to understand the bigger process. That's what I would suggest. So I hope this has helped. Next, we will look at the Krebs cycle and its role in the process of ATP production. Good luck, guys, and remember, if there's any questions, ask me.